Since we do go to some trouble to make the videos available to you, I occasionally look at the statistics to see if anybody actually is looking at them. And I noticed something interesting about them, which is there is a lovely large number of people who do use them to justify the effort, but tends to be focused in the week before an exam. Better late than never, but you will probably find them more helpful if you look at them sooner than the week before the exam. Now, you're here, so you actually probably don't need to be told this. But perhaps the people who are going to listen to the videos who didn't come will hear this, hopefully before the week before the exam. And in fact, I might even send them a little email saying, you might want to look at it before the week before the exam. In any case, I am glad to see people look at them. I do care. I've gotten one email about some difficulties with the video. I tried to reproduce those difficulties and couldn't do it. Are other people having generally problems with access? Yes? On the Mac, are you using the Google link or icon? I don't believe anybody succeeds in making icon work for Apples, but the Google links are there precisely because I'm an Apple person. So if you go to the second toolbar, the one that's like colored green or something, the links is right at the right-hand side, just about. The links is where you'll find the Google videos. Any other issues, problems? Someone couldn't download the Google video. I tried, I did. I don't know where the problem is. Sometimes it takes more than one try. Other issues? Okay. Well, let's do real biochemistry. That's always more interesting. So we were talking about photosynthesis, and we had sort of introduced the topic and gotten through what we call the light reactions. So in the presence of light, we can have photo events happening. This is slide number 3 on your handouts. In the presence of light, we can have photoabsorption by either photosystem 1. Come back here, there we go. Options. Either photosystem 2 or photosystem 1. Both of these have pigments that absorb light and allow for photooxidation and reduction. The electrons that are excited through that process are transferred through an electron transport chain, somewhat similar to what happens in oxidative phosphorylation. Cytochrome B6F is an intermediary along the way, takes electrons from plastoquinone, transfers them to plastocyanin. And then in the process of this, here and here, protons are moved. We create a proton gradient and then use a structure very similar to the ATP synthase of mitochondria to exploit the proton gradient and make ATP. So that's sort of the grand overview of that. And then we need to get into the dark reactions. They're only dark in the sense that they don't require light per se. What they require is the products of light. So the dark reactions are going to... people have any questions about linear versus cyclic electron transport, that distinction? The zigzag pattern versus going around and around with just photosystem 1? Okay. So we can then move on to the dark reactions in which we use the products of the light reaction. And the products of the light reaction that are relevant here are ATP and reducing power in the form of NADPH. And it's going to be possible through the enzyme Rubisco. Rubisco is the nickname for ribulose bisphosphate carboxylase, adds a CO2 to ribulose bisphosphate. But unfortunately, it is not a perfect enzyme. It has the capacity to add oxygen instead of carbon dioxide. And so if you have, come back. Arrow options. So if you have ribulose bisphosphate here, if we add oxygen to it instead of adding carbon dioxide to it, it will split the molecule, but one of the two products now will be a 2-carbon fragment instead of a 3-carbon fragment. And this 2-carbon fragment, 2-phosphoglycolate, is not useful by itself. It can be restored to usefulness, but it's going to cost energy in the form of ATP, and it will have to be reduced, so it will consume some of the NADPH. And so this is an example of oxygen consumption in the presence of light, and it's called photorespiration. You and I do respiration, that is, we consume oxygen and give off CO2. Photorespiration consumes oxygen in the presence of light. It's a wasteful process. Now that we've got some carbon fixed, let's assume that Rubisco did the right thing, added a carbon dioxide. We now have some molecules of 2,3-phosphoglycerate. This should read 2 molecules of 3-phosphoglycerate, not 2 comma. Okay. So here's the fundamental reaction. The ribulose 5-phosphate is going to be phosphorylated in order to make it ribulose 1,5-bisphosphate. And here's this key reaction by Rubisco in adding the carbon dioxide. And when split, you have 2 identical molecules of 3-phosphoglycerate. This reaction, this fixing of carbon, is something that happens in the stroma of the chloroplast. The stroma is kind of the meaty interior of the chloroplast, and it's analogous to the matrix of a mitochondrion, where the sort of the mass of the enzymes is kept. You might wonder how you could track something like this down. And it turns out that you need to have radioisotopes to be able to do it. And radioisotopes were not available to us until roughly World War II. And during World War II, Berkeley, University of California at Berkeley was one of the few places that had a cyclotron and could make radioisotopes more or less on demand. And the isotope that they were able to make first and most easily was something called carbon-11. And carbon-11 is very hot, but it's also very short-lived. And so in the early studies of this reaction, the cyclotron would irradiate carbon and make carbon-11. They would purify it as quickly as they could, and a brave graduate student would get on a bicycle and race down the hill to take the carbon-11 to the biochemistry lab, where they would run an experiment or two before the carbon-11 died. Life is a little easier now than it was then. So once this reaction has happened, we do have now carbon that was in gas now fixed into an organic molecule. What do we do from here? Well, there are a number of different reactions that are going to have to happen, and you're going to probably recognize them because they do resemble a number of reactions you've seen already. Some of these reactions resemble gluconeogenesis, which makes perfect sense. Gluconeogenesis was making sugar out of 3 carbon fragments. That's what we need to do here. So we have the 3-phosphoglycerate, which we made using Rubisco. Phosphoglycerate kinase, we've seen that one before, consumes one of the ATPs we made in the light reactions. We now have 1,3-bisphosphoglycerate, which you probably recognize from gluconeogenesis. Here we have 1,3-bisphosphoglycerate, and we can consume some of the NADPH we made and reduce it down to glyceraldehyde 3-phosphate, splitting off an inorganic phosphate in the process. So this is the opposite from what we saw in glycolysis. It's the direction we need to go in gluconeogenesis, but in the chloroplast, when we're looking at the Calvin cycle, we're going to use NADPH as the donor. Okay? So this is an isozyme, if you like, of the gluconeogenesis enzymes. So now we have a molecule of glyceraldehyde 3-phosphate. It's been reduced on this carbon, right? We had a bond to an oxygen that's now a bond to a hydrogen. So in general, anabolism is going to try to reduce things to make them more and more exciting. And what do we do from here? I want to give you kind of an overview of what's going to happen before we try to look at it in a little more detail. And this diagram from your textbook is actually one of the more intelligible diagrams I've seen of the Calvin cycle. You'd think that something that's the center of life should be a little easier to understand, but it seems to be hard to represent to people. Now, when we were doing glycolysis for the second half, we talked about 2 copies of glyceraldehyde 3-phosphate at each step. So to make this one a little easier, let's start off with 3 molecules of ribulose 1,5-bisphosphate. So let's start off with 3 5-carbon sugars. We've got 3 5-carbon sugars. We're going to fix 3 molecules of CO2. And since each time we fix a molecule of CO2, we split that 5-carbon sugar into 2 3-carbon pieces, now we have 6 copies, 6 molecules of glyceraldehyde 3-phosphate. We're going to take out one molecule of that glyceraldehyde 3-phosphate and use it to make other things. Make glucose, make amino acids, whatever. The other 5 pieces of the other 5 molecules of 3-carbon sugar, we're going to rearrange in order to get back our 5-carbon sugars. This should sound a little bit like something we've seen before, which is the pentose phosphate pathway, where we could mix and match 2 and 3 carbon pieces to change the length of the chain and so on. So the overview of what we're going to do is we're going to start with a 5-carbon sugar, fix one molecule of CO2 gas to it, have 6 3-carbon pieces, or 6 times 3 is 18 carbons. We pull out 3 of those carbons for anabolism in general, and then we recycle the other 15 carbons to get back to where we were before. And we can do this over and over again as long as we have ATP and NADPH. So that's the overview of where we're going. So a pathway like that, which is, it's fairly expensive. It's taking ATP, it's taking NADPH. You would want that thing to be only active at the right time, when you had the raw materials you're going to be used for that. And in fact, there are a number of steps at which all of this can be regulated. One molecule that you would expect to be regulated is Rubisco, and it has a site in which, on the enzyme, on the active site, where the carbon dioxide has to bind to the enzyme to be added on to the ribulose 1,5-bisphosphate. That activation site is favored at high pH. Now, since during photosynthesis, you're pumping protons out of the stroma, the pH of the stroma is going to rise the more active the electron transport of the thylakoid membrane is. So high pH is, chemically speaking, the same thing as saying photosynthesis, the light reactions are rolling fast. This is the site on the enzyme itself, which is where something's going to be happening. Here's a lysine side chain where we want to have the carbon dioxide become covalently attached. And since in the process of adding the carbon dioxide to it, that gives off a proton, that's a reaction that by mass action will be favored when the pH is high and protons are less abundant. Another way in which Rubisco is regulated is through magnesium ions, and the magnesium ion concentration in the stroma also rises during the light reactions. There are a number of other enzymes that are involved, and many of them are regulated through ferredoxin. And you remember that ferredoxin was the electron receptor that was going to be reduced by the electron transport chain in the light. And this gives you a little bit of a feeling for some of the enzymes that are regulated through ferredoxin. So ferredoxin can, in its turn, reduce a smaller molecule called thioredoxin, and thioredoxin plays this role in the regulation. Here we have fructose 1,6-bisphosphatase, which we recognize as being gluconeogenesis. Glyceraldehyde 3-phosphate dehydrogenase is going to be present in a number of different pathways. Acetoheptulose bisphosphate is a 7-carbon phosphate splitting enzyme. It's in the pentose in the Calvin cycle, and so on. This last one is a little bit different. NADP plus malate dehydrogenase. We're going to see something called the C4 pathway, that there's an alternate way in which carbon dioxide can be fixed, and that gives certain plants, such as corn, an advantage over the simple, plain, bare bones approach to carbon fixation. This is not in your textbook. Let me just take a peek and see if you can read it. Yeah, you can read this off of yours. Good. It even looks good on the screen. This is, to my mind, the clearest, most elegant representation of the Calvin cycle that I've ever seen. Excuse me. I so rarely get phone calls. I didn't think to turn it off. I apologize. One of the things that I like about this is that it puts in place all of the compounds of the Calvin cycle in a way that makes it clear it's a cycle. Another thing that I like about this diagram is that it does a good job, I think, of showing you stoichiometry, how many molecules of things are used along the way. And so let's just walk through this a little bit to have a sense of it. We've mentioned that we have to start with a 5-carbon sugar. Here's ribulose 5-phosphate. It costs us ATP to make it into ribulose 1,5-bisphosphate. So we're going to do that 3 times. So it's going to consume 3 ATP. Now we have those 3 5-carbon sugars that I pointed out. We're ready to fix 3 molecules of CO2. And now we have 6 molecules of 3-phosphoglycerate. We're going to consume more ATP to make 1,3-bisphosphoglycerate. Again, sounds familiar from gluconeogenesis. And to take that to glyceraldehyde 3-phosphate is going to need reducing power, and the reducing power is in the form of NADPH. This is a purely anabolic process, and it uses NADPH. And we just throw away the energy of that inorganic phosphate. One of those 6 3-carbon fragments can be pulled off. They're, in this case, pulling it off as dihydroxyacetone phosphate. That's not so important. The other 5 have to be put back together and rearranged to get back up to 5-carbon sugars. And what's going to go on here should look familiar to you from pentose phosphate pathway. I don't expect you to be able to draw the whole pathway, but walk through it with me so you can see the highlights and appreciate what's going on. So 2 of those molecules, total of 6 carbons, we can make fructose 1,6-bisphosphate. Again, aldolase condensation. We can throw away one of those phosphates and have fructose 6-phosphate. We can take a 3rd molecule of GAP and smush that on top of the 6-carbon fructose. So we have altogether, it would be 9, but this enzyme is going to split it and rearrange those. And so what we have is erythrose 4-phosphate and xylulose 5-phosphate. So 4 and 5 is 9. We rearrange the 6 and 3 to 4 and 5. The xylulose 5-phosphate is already a 5-carbon sugar, and it's fairly easy to make it into ribulose 5-phosphate. So we've got one of our ribulose 5-phosphates back. Here we have the other fragment of this, the erythrose 4-phosphate. It's so handy that they're phosphorylated on the end. You always know how long they are. And here's dihydroxyacetone phosphate, so a 3 and a 4. We now have cetoheptulose 1,7-bisphosphate. It's a heptulose. It's a 7-carbon sugar. You're not going to find this on the grocery store. But it's a bisphosphate, so it's a long 7-carbon sugar, phosphate off on each end. And a phosphatase is going to split off that, so we now have cetoheptulose 7-phosphate. We have 1 GAP left here. So if we take that last one and smush it onto the cetoheptulose, 3 and 7 is 10. We can split that into 2 5-carbon sugars, ribulose 5-phosphate, xylulose 5-phosphate. These are both of them 5-carbon sugars. Pretty easy to rearrange them into ribulose 5-phosphate. So now we have back 3 molecules of a 5-carbon sugar. So it's a really elegant pathway. I hope you hear as sort of a rhyme the overlaps with things like the pentose phosphate pathway and a couple of reactions from glycolysis and gluconeogenesis. So, any questions about this before we go on? If you're curious about that phone call, it was probably that Jetta that someone asked me about it. Turns out that it had very bad alignment. So the carbon fixation that we've talked about so far through Rubisco, the classic model, is sometimes called C3 fixation because the smallest molecule or the first molecule you find the carbon in is a 3-carbon fragment, glyceraldehyde. C4 carbon fixation takes a slightly different way of doing it. And because of the ways it's changed the process, it makes carbon fixation more efficient when it's hot or dry or both. And it's called C4 because the first product is a 4-carbon fragment, and it can happen in the dark. The Calvin cycle, in principle, can happen in the dark. We call it dark reactions for that reason. In a C3 plant, usually it's going to happen in the light because that's when the ATP and NADPH is made. We're going to see there's some C4 plants that actually do their carbon fixation in the dark. It's not a universal pathway because it's more expensive. It costs extra ATP and extra NADPH to do C4 fixation instead of C3. If you've got a lot of light, maybe that's not a problem. Remember, each of us makes our weight in ATP every day. So if you have a source of energy, ATP is cheap. Another cute trick that C4 plants sometimes take advantage of is to separate in space the dark reactions from the light reactions. And this gives them certain efficiencies. And I've already mentioned that corn is a C4 plant, as is grass, which is why grass grows so well, even when you don't want it to. So let's look at some of the C4 reactions. We're going to see some things that resemble other pathways, so that it won't be completely new stuff to learn. It will use NADPH because these are anabolic. And we're going to regenerate phosphenylpyruvate in a way that allows us to overall capture carbon dioxide. So if we've made some phosphenylpyruvate through our usual reactions, we know that that's going to cost some energy, right? There's 2 reactions to get from pyruvate back up to phosphenylpyruvate. Here's a slightly different enzyme, phosphenylpyruvate carboxylase. And this is using bicarbonate here, not CO2. Carbonic anhydrase generating bicarbonate. Oxaloacetate. We're going to generate malate from the oxaloacetate, so that's the backwards direction from the citric acid cycle. But we're using NADPH. So this malate dehydrogenase is a different enzyme, chloroplast, anabolic using the pH. The malate is now available to produce a carbon dioxide through a special enzyme called malic enzyme. And in the process, it will reduce NADPH again and regenerate the pyruvate. So this is a way of capturing carbon dioxide sort of temporarily, if you like. These are not the endpoints. These are a way of capturing some carbon dioxide for a while. And then we're going to feed the carbon dioxide we captured through these reactions into an ordinary, plain old Calvin cycle set of reactions. So how does that work out in practical terms? Here's the phosphenylpyruvate carboxylase reaction. We're going to, this particular enzyme can simply use the bicarbonate. If you remember, in gluconeogenesis, we started from oxaloacetate and made phosphoenolpyruvate. That was a different enzyme. That was phosphoenolpyruvate carboxykinase, which was named for the wrong direction. And so what we've done is to extend that 3-carbon backbone into a 4-carbon backbone. Since we lose this phosphate, this is a high-energy phosphate, if you like. We lose the phosphate as we add the bicarbonate. That costs us 1 high-energy bond, in effect. Here's the malate dehydrogenase. It's named, in fact, for the backwards reaction, and again, uses the NADPH for anabolic purposes. You may remember this enzyme also, or this set of reactions from the mitochondrial shuttle. But in the mitochondrial shuttle, we were trying to shuttle NADH from the cytoplasm into the mitochondrion. This is a little different, and here we're doing it in a single compartment and using NADPH as the reducing, NADP plus as the oxidizing agent. And here's the carbon dioxide that we need for the Calvin cycle. Sort of like, what's the point, right? It makes more sense if we look at it in the context of the anatomy of C4 plants. So here is one typical way that a C4 plant would organize these metabolic pathways to take advantage of this process. So this is one very good example of metabolic compartmentation, that sometimes where you do what you do can be as important as what you do. And so here in a typical C4 plant, you have 2 kinds of cells, a mesophyll cell and a bundle sheath cell. The mesophyll cells are just kind of loose filling in type cells that fill in the interior of a leaf. And the bundle sheath cells are more specialized cells that are kind of a little bit inside relative to the mesophyll cells. So the carbon dioxide has to get into those cells. It's going to be let in through structures called stomates. And the mesophyll cell can absorb the carbon dioxide and combine it with the phosphoenolpyruvate to make an oxaloacetate. It will then reduce that to malate. The malate can then move physically. It is actually transferred from the mesophyll cell to the bundle sheath cell. And in the bundle sheath cell, the carbon dioxide is let loose for the Calvin cycle. The pyruvate produced in that is sent back to the mesophyll cells. You spend some ATP to regenerate the phosphoenolpyruvate. You can see this is a different reaction than the one we saw in animals. We're taking ATP and an inorganic phosphate off. So it still costs 2 high-energy bonds, but it's in a single-step reaction. So now we have phosphoenolpyruvate and can do this all over again. So what this does is it allows the mesophyll cells to be exposed to the carbon dioxide and move that fixed carbon over to the bundle sheath cells, where the bundle sheath cells then have the carbon dioxide given to them. And the advantage to this is you're protecting the bundle sheath cell from exposure to oxygen. That it is at a distance from the rest of the atmosphere, and the oxygen in the atmosphere has to pass through all the mesophyll cells first. So the Calvin cycle, which is running off of rubisco, is not exposed to the same concentrations of oxygen that the other ones are. So it reduces photorespiration. That's one thing that makes it more efficient. The other thing is that in principle you could generate enough carbon dioxide here to raise the concentration of carbon dioxide relative to the atmosphere. So one of the reasons that not every single plant bothers with this is it's costing us 2 high-energy bonds to move that carbon dioxide from the mesophyll cells over to the bundle sheath cells. There's another family of plants called the crassulaceans that do take advantage of this C-form metabolism, but they do it over time, not space. So they open up their stomates at night when it's relatively cool and not as dry. They do a lot of carbon fixation at night, and then they close their stomates in the daytime, and they do their light reactions and take advantage of all the carbon dioxide in the daytime. If you've ever had rock plants or various kinds of really big, fat, succulent-looking things, many of them are crassulaceans. And it's an adaptation, again, to dry and hot. Any questions about this before we go on? Okay. So we've done our Calvin cycle thing. We've done gluconeogenesis. We've got glucose. What do we do with it? Well, one of the things that we can do with it is we can make starch. And we can't make starch just out of raw glucose, of course. We need to activate it. So a glucose 1-phosphate and adenosine triphosphate can give you ADP glucose. You may remember UDP glucose from before. Plants, when they make starch, use ADP glucose and an inorganic phosphate to drive the reaction. ADP glucose is an activated kind of glucose that's a good substrate for the starch synthase. So if you have a partial starch chain, the ADP glucose then can be added onto the end of that, releasing the ADP to be reused again. So it costs, on average, 1 high-energy bond to add a 1 glucose to a starch molecule. So you have all the energy of the glucose synthesis plus 1 to make it into a stored form as starch. There are some other sugars that are of importance, of course. Sucrose is one of those. I think I may have mentioned that sucrose is one of the major sugars in plants, and we think that may be because sucrose is not a reducing sugar. The way it's put together, there's no group on it that attacks other things non-enzymatically. And how do we go about that? So here we have a UDP glucose, which we saw earlier when we were talking about a mammalian carbohydrate metabolism. Here's a fructose 6-phosphate. Could have come out of the Calvin cycle or out of gluconeogenesis. Both of these now are moderately activated, and the enzyme that synthesizes sucrose then can form a bridge there. We now have sucrose 6-phosphate, removed the UDP from the glucose, and hydrolyze off the phosphate to have plain old simple sucrose, which is this glucose-fructose disaccharide. And this linkage here, this alpha 1,2 linkage, is an extremely inert linkage compared to some of the others in the sugars. This intermediate, the UDP glucose, which is one of several activated forms of glucose, is also the precursor for cellulose, which is kind of interesting. They use ADP glucose for the starch, UDP glucose for the cellulose, almost like the NAD, NADP distinction in reducing power. The thermodynamics are identical between ADP and UDP glucose, but the enzyme that uses it, of course, cares a great deal about which one it is. If I've left you with the impression that only plants do things that are interesting with light, that would be mistaken. There is photochemistry in light. And in fact, most animals, down to quite simple ones, have some kind of light-sensing molecules, which means they have to have some kind of structure that will absorb those light photons. One of the common things that is light-driven are the circadian rhythms. Circadian stands for circadian, around a day. Our bodies will naturally cycle on roughly a day-length cycle. If you put people either under continuous light or continuous dark, they get awake and sleepy at roughly 24-hour cycles. It's a little bit longer, like 25 hours, which is a nuisance. Light entrains that cycle, which is one reason that all human beings need a certain amount of real daylight. We call these things cryptochromes because we're not completely sure what the sensor for that system is. There are some components that seem to be involved, such as flavins. Here is a flavin, and you can see this resonant structure within the molecule, very good for picking up photons. Here's a pterin. Again, you can see resonant structures that are very good for picking up light. The pterins tend to transfer their light energy to the flavins. This is a diagram that I've added to those in the textbook. Obviously, your handout is not going to be brightly colored, but if you want it online, it will be available in a colored form. And I picked it because it's going to have an analog on the mitochondrial transport chain, so you can get a good sense of the similarities of the two things. This is the photosynthetic electron transport chain laid out for the Z scheme when you have linear electron transport. Excuse me. Here's your photosystem 2 absorbing light, transferring protons, and generating oxygen gas. The electrons that were generated in that reaction are transferred to plastoquinone, lipid-soluble carrier, which passes on to the cytochrome B6F that has iron-sulfur centers and various other good things. Redox reactions in it also pump protons, and the electrons end up on plastocyanin, so it's a peripheral membrane protein like the cytochrome C, and it will carry electrons then to photosystem 1. Photosystem 1 can have another photooxidation reduction event with light interaction, and the electrons from plastocyanin then restore the cytochrome P700, the chlorophyll P700. Electrons in the linear scheme, when you're trying to generate NADPH, then are transferred to ferredoxin, and the ferredoxin then can be used to reduce NAD plus to NADPH. The protons here and the protons there flow back through the ATPase, you make ATP, and this is in the stroma. This is the lumen of the thylakoid. Typo here, that should be an A. And what's rather nice, and I have not seen this very often, is the color coding between the physical structure of the complexes in the membrane and the position on the redox scale of the different components of the system. So that may help a little bit also in sort of mentally tying together the physical things and the redox potential. And then the nice part of it is that then it's also available to show you the analogous reactions that are happening in mitochondrial membranes. So you can see again complex 1, it's not getting its electrons through photosynthesis but from NADH. Those are again generating a lipid-soluble carrier, ubiquinone, not plastoquinone. In this case they're going to complex 3, protons being moved in both cases. Here's cytochrome C, the peripheral membrane protein that's the analog of plastocyanin, transferring it to complex 4. And complex 4 consumes oxygen instead of producing oxygen. And then again, an ATP synthase. The only thing that would make this pair of diagrams more perfect is if they got the topology right, because here the intermembrane space is at the top and the matrix is in the bottom, whereas in the chloroplast it was the other way around. So just be aware that they did flip the geometry in that case. But in both cases, it's a proton movement. This should be in your handouts as a full-page thing, and I don't require anybody to do it, but some people think better with their hands than with their eyes or whatever. And this is a color your own chloroplast, if you like. A couple of people have commented that they found it helpful. I don't tell anybody that you have to do it, but if you think this will help you, I certainly offer it to you as a way to fix in your head the relationships of what's going on. Any questions about this exercise that's available to you? You can play chloroplast for a day. Okay. The other full-page thing that I hope you have. Oh, what? Oh, maybe it's on. I think I know what it is. All right, so it's not in this. Let me just open up one more. No, that's not mine. There should be. I know there's another page to yours. There is, and I will get my. Go away. No. Okay. Here we go. This is it. Okay. Let's just do it here. So if you turn to your last page, let's try it just like this. So let's start with the overview aspect of it first. This is a metabolic map unlike any that I've ever seen before. And since we've just finished the photosynthesis aspect of metabolism, I thought this might be a good time for you to get a chance to take a peek at it. Some of you may remember my mentioning Prochlorococcus back near the beginning as the smallest free-living photosynthetic organism, that this provides like 20% of the oxygen in the atmosphere. It's about a half a micron across. And this little blue-green alga, it's a marine bug, has, because it's free-living, all the things it takes in order to be a free-living photosynthetic organism. But what's unique about this metabolic map that I hope will be helpful to you is that it puts the metabolic pathways where they are in the cell. It's a small enough cell we can fit them in. And so you can go around and see the various transporters here. And I think, yes, you can see on your handout the actual names of these things. I'm not expecting anyone to memorize the map. Please don't worry about that. It's meant to give you a sense of how these things are fitting together. It's a very sparse organism. The only nitrogen it can take up is ammonia. So other plants will take nitrate or nitrite. It only uses ammonia. And here's its one singular ammonia transporter. It has transporters for a number of other metals over here, like iron and zinc and so on. It can pick up, there's some more iron, some more inorganic things here. It can pick up di- and tricarboxylic acids. So it's just this kind of promiscuous, is it an organic acid? Yes, come in. Here's a bicarbonate exchanger, potassium pump. So a much more minimal set of transporters than your cells or mine. You can see here, yes, TCA cycle, which of course it isn't a cycle. You see there's a big hole missing there. This is the classic incomplete TCA cycle of the blue-greens, but you can see on it how it's useful down this direction and useful down that direction. You can see up in here the Calvin cycle and the reactions we were talking about today and how they connect to make, ultimately, glucose. Here's glucose coming out of the Calvin cycle. It doesn't go into all the detail of the amino acid pathways. It just gives you a general sense of how things get into those pathways. We haven't started nitrogen yet. You may want to revisit this when we've done that, but it shows you how you pull things out of the TCA cycle to make these amino acids, how you pull out pyruvate to make these amino acids, and so on. And then, quite charmingly, it has the photosynthetic membranes over here. So you can see that they are physical, that gene products are physically organized together. So it's got photosystem 2, which is obviously more complicated than our diagrams were. It has cytochrome B6F here, and photosystem 1, and the ATP synthase. This, of course, should complete because it's a lumen, not a broken end there. And if you look here right between, NADH dehydrogenase. So the efficient way this little teeny bug copes with life is it includes an NADH dehydrogenase in its photosynthetic membranes and just shoves the electrons from NADH into the same electron transport chain with the electrons that it's getting from photosynthesis. It's a very cute trick. So I thought you might enjoy seeing how these things literally fit together in an organism. I hope it'll be helpful for you to see how they tie together, and I hope you all have a good weekend.